to the cloud. Okay, I might be recording now. So I'm share device is on, so PowerPoint back on. Okay. All right. So I'll start from here. PowerPoint. All right. Can you guys see this? Hello?
Yeah.
Okay. All right. Perfect. Okay. So this is where we ended yesterday. We talked about the platelet system and we talked about the coagulation system, right? And I did mention that there's so many little factors that have to take place. And again, it's not two separate events, but it's happening. There are systems that are interrelated, and they're happening at the same time. So with that said, now that we've talked about the platelet system, we talked about basically primary hemostasis. So you'll hear people say, or the literature say, either primary hemostasis or the platelet system, and we just have to, make sure that we know that we're really talking about the same thing. So they call it the platelet system because platelets are involved, right? And really it's talking about all of the functions that the platelets have in primary hemostasis. But I don't want you to forget that the platelets also function in secondary hemostasis or in the coagulation system as well. They're a huge part of the coagulation system, okay? So as platelets are adhering and getting activated, they will promote, these changes will promote shape changes, and it'll provide a phospholipid surface. And we need this phospholipid surface so that fibrin clots can form on top. Okay, and remember, during the activation process, there's a translocation of the polar phospholipids from the inner to the outer membrane, and we need these polar surfaces so that platelets aggregate and coagulation factors can form complexes on these platelet surfaces, these phospholipid surfaces. Okay, also other receptors on the surface of activated platelets will generate thrombin. And remember yesterday I asked you to make a big circle around thrombin because it's going to be super important. Also to keep in mind, platelets, again, they're these little cytoplasmic structures, and they don't have a nucleus, but they have organelles that are very important, right? Can anybody remind me of what those organelles may be in the platelets?
Organelles.
Yeah, remember we talked about yesterday some organelles that are in the platelets that are very, very essential in the coagulation process. Anyone remember? Take a guess.
Mitochondria.
Right, well, dense granules, right? And alpha granules, and these secrete substances, right? So we need those substances for during platelet activation in order to promote platelet aggregation so that we can stabilize that platelet plug, that initial platelet plug, okay? Okay. So let's see. Okay. So we don't need that. Hold on. Most coagulation factors exist in plasma, and when they are in the plasma, they're inactive. Okay, so they're called zymogens. And then they become activated enzymes or proteases. Each coagulation factor will activate another factor, and this happens in a very, very carefully controlled sequence. It's called the coagulation cascade. And during this course, you're going to be very, very familiar with this coagulation cascade. It's very similar to a chain reaction. The factors that we'll be dealing with and talking about have individual names, which we'll learn about, but we refer to them by Roman numerals. Okay, so remember yesterday I was going through factor 5, factor 8. So they all have names, but we're just gonna call them by the Roman numerals because that's the more I guess the more popular form. Okay, the ultimate goal of the cascade is to make thrombin, okay? And thrombin essentially converts fibrinogen to fibrin. Fibrin is the actual stabilized clot, okay? All right, so here are some of the actual names, their common names for the factors that we're going to be talking about. And factor 1, we never really call it factor 1, we just call it fibrinogen, okay? Same thing with factor 2, the common name is prothrombin, okay? TF stands for tissue factor. Tissue factor is not normally circulating in the blood, but it's found in surfaces of tissue factor bearing cells. And these can be fibroblasts, they can be smooth muscle cells in the subendothelium, and other extravascular cells. Factor 5 is labile factor. Factor 7 is called the stable factor. Factor 8 is called the anti-hemophilic factor. Factor 9 is called the Christmas factor. Factor 10, the Stewart power factor. 11 is the plasma thromboplastin antecedent factor. Factor 12 is the Hageman factor. Pre-kallikrein or pre-K is the Fletcher factor. And high molecular weight kininogen is HMWK. We'll use that, HMWK. And then factor 13, you have the fibrin stabilizing factor. All right, so some important characteristics about these factors is that all factors for coagulation are synthesized in the liver. Factor 5 and factor 13 are also believed to be synthesized in the megakaryocytes. And factor 8 is also believed to be synthesized in other sites other than the hepatic cells, such as endothelial cells, okay? Storage labile are factors 5 and 8. This means that their ability to function may be altered depending on the storage. So if you don't store the blood in the correct manner, you're not going to get the correct results if you're testing for factor 5 or factor 8, okay? Factor 5 is unstable in EDTA, and fibrinogen and factor 8 are both acute phase reactants. Do you guys know what these are, acute phase reactants?
I do not.
No. So acute phase reactants, and these they can be proteins, they can be factors like this one, that there may be found in normal levels in the blood or in circulation, but whenever there is an inflammation, right, they go up in number, okay? So we have to keep that in mind because sometimes we're trying to rule out certain conditions, but if a patient has an underlying condition that it's perhaps inflammatory, right, those factors are going to be increased anyway. So we cannot confuse a deficiency in factor 8 or factor 5 when someone is going through an inflammatory process, okay? Because those factors are going to be high anyway. So these are called acute phase reactants. And there are other reasons why they get increased, right? It can be due to inflammation, so some condition that is causing inflammation in the body, or it can be pregnancy. Sometimes taking oral contraceptives also triggers these acute phase reactants. And instances like having cancer does as well, okay? So fibrinogen, which is factor 1, and factor 8 are two factors that are acute phase reactants. So we have to watch out when we are assessing these factors. Okay, fibrinogen or factor 1 is the one factor that it is in the highest concentration in the plasma. So you'll find fibrinogen in is the most abundant factor. The one that has the shortest half-life is a factor 7. Do you guys know what that means? Having a short half-life, plasma half-life?
They don't live long.
Yeah, so it doesn't live, its half-life means their ability, it's basically the lifespan of the factor or of the protein, right? And it's 50% capacity of that protein or the factor. So it just so happens that compared to the other factors, factor 7 has the shortest half-life, okay? So that's something to watch out for, and I'm going to explain that later on when we talk about testing. Factors that can get activated at cold temperature, factor 7, it's another one, okay? So basically what it means is that if I collect a Coag sample, okay, and I am testing for factor 7, okay, I cannot put this sample in the refrigerator because cold temperatures are going to activate my factor. If my factor gets activated, that means it's going to start working, right? So then that means that when I'm testing for the time it takes to clot, factor 7 has already been depleted, okay? So that means that my times to clot are gonna get shorter. And this will make sense, it'll make more sense a little bit later, but activated temperatures are an enemy for factor 7, okay? So we have to keep that in mind when we're testing for factor 7, okay? So factor 7, two things. It gets activated at cold temperatures and it has the shortest half-life in the plasma. Okay. Okay, moving on, let's see. Now, do you guys remember the difference between plasma and serum?
Plasma is serum with formed elements, and serum is plasma without the formed elements.
What do you mean by formed elements exactly?
Red blood cells, white blood cells, platelets.
Right. So when you because serum will obtain it, right, in a red tap tube, right? And what we're gonna do is we're gonna allow that to clot, okay? So we're in the serum, right? What you were saying is we're not gonna have some of the proteins, some of the coagulation factors in the serum. In the plasma, however, you would collect that in a sodium citrate tube, which is the blue tap tube. And when you spin it down, all right, you do have those elements in there, which would be your coagulation factors, right? So you'll have the factors that you're looking for in the plasma. So again, serum, you allow it to clot, and then it's the liquid part of the blood. And then plasma, you spin it down, okay? And in the plasma, you will find those coagulation factors, okay? So this is why it says here, these factors will not be found in serum, okay? So why do we have to make that difference in coagulation? Why is it so important to make sure that we identify that difference? In Coag, what we're trying to do is we're trying to identify the activity of these factors, right? So if we run a serum that doesn't have fibrinogen, prothrombin, factor 5, factor 8, factor 13, we're not going to be able to use them for our coagulation testing, right? Because remember, in coagulation, there's a whole coagulation cascade, and all of these factors have to come together to form the clot. These are the coagulation proteins that are going to form on top of those platelets that we were talking about, okay? So if we don't have it, we use serum, and we don't have these factors, we're never going to get the correct results, okay? Therefore, we use plasma, okay? Okay, and some chromosomal locations for certain factors are important to note, okay? Because some deficiencies will come from genetic factors. So genes for factor 8 and factor 9 are located in the X chromosome, And genes for the other factors are located in the autosomes, which are just your typical chromosomes. The X chromosome is the sex chromosome, right? So that's going to make a big difference because these conditions, right, hemophilia A and B, have to do with factor 8 and factor 9, and these are passed down from parents, from your from the X chromosome. Okay, so this is a picture from your book. These are all the little proteins and factors and enzymes that we're gonna like really get to know well during this course, and make it a little bit complicated. But when you do it over and over, you'll start remembering. So it's a little bit of memorizing here in Coag. Okay, all right. So so we talked about some of the cymogens, right? Which means that they are circulating in plasma in an inactive form, right? So cymogens turn into enzymes, okay? Then you have some cofactors, which are high molecular weight kininogen. You have factor 8, factor 5, tissue factor, protein S, thrombomodulin, and protein Z. Then you have control proteins. Control proteins are proteins that are regulating coagulation. So basically, they're either enhancing coagulation or they are inhibiting coagulation in some sort of way. And they all work a little bit differently, okay? So these are control proteins. So we need to have these control proteins in place because if we don't, right, what would happen if we have factors that are constantly coagulating? So if that happened, we'd be forming clots all the time, right? So our body has a way of stopping these clots and reversing them. And that's what we talk about. That's why we have control proteins. These are controlling these factors. So clotting doesn't occur all the time, okay? So the formation of a fibrin clot requires a series of enzymatic reactions, and that involves your coagulation factors. Okay, so these are all special biochemical groupings. What you're seeing are biochemical groupings of coagulation proteins, okay? You have your proteases, you have your cofactors, and your control proteins. All right, so right here, this chart just shows you, this is again from the book. It gives you the function of each of these cofactors. Tissue factor is a procoagulant. So that means that it's going to enhance coagulation, right? It's going to call for coagulation. And it mainly binds activated factor 7. So do you guys see that factor 7 right there? And you see where it says a little a? It has a little letter A next to it. What that means is that this has been activated. Factor 7 has been activated. When you just see factor 7 without the little a, that means it has not been activated yet, okay? A cofactor, factor 5 is a procoagulant too, and factor 8 is a procoagulant, okay? And they bind factor activated factor 10 and activated factor 9 respectively. High molecular weight kininogen is another procoagulant, and it binds activated factor 12 and prekallikrein. Thrombomodulin is functions as a control protein to bind protein C, and also functions as an antifibrinolytic protein, which we'll discuss in a little bit, or TAFI, and it binds thrombin. Okay. Okay. So what would antifibrinolytic mean? Any ideas?
Is it like to stop clotting?
Remember what fibrinolysis means? What does that mean? Fibrinolysis. Think about it,
Break
guys.
down the fibrin.
Yeah, you break down the fibrin clot, right? So now we're looking at something that's called antifibrinolytic, right? So now the body has a way of breaking down the clot, but now we have a protein that stops that. Okay, so again, it's going to be a little bit complicated. There's a lot to remember, but we need to have these proteins in place, and I'm going to explain that and how they work. Okay, antifibrinolytic. Okay, protein S and protein Z are also control proteins, and they bind to protein C, tissue factor pathway inhibitor, which is TFPI, and ZPI as well. Okay. Right here, this is, and this should have been the slide that we were talking about before, the difference between serum and plasma. Okay, so sodium citrate 3.2 is the anticoagulant that we use for routine coagulation testing, okay? It is, the sodium citrate is the additive that is actually in the tube of the light blue stopper, okay? So remember we talked about, well, we're gonna talk about this coming week too, about these vacutainer tubes and what they have inside, but you may remember some of it from the video that we looked at the other day in phlebotomy. So this anticoagulant, it's really just small amounts, okay? And it's sodium citrate 3.2. Sometimes they use sodium citrate 3.8, and that's okay, okay? So the function of sodium citrate is to bind calcium, okay? And what we want to do with this anticoagulant is we want to prevent the blood that we're collecting from clotting. That's why it's called an anticoagulant. So the way it functions is it's going to contract, it's going to try to bind all the calcium elements in the blood, because calcium is needed for what?
The
Clotting.
clotting factor.
Yes, you need calcium for clotting. And so calcium is going to promote clotting, right? But when you bind it, that's not happening, okay? So it helps in the anticoagulation of the blood. So we don't want the blood to coagulate when we're doing coagulation testing, because if it coagulates, all the platelets are going to be consumed in the clot, and then all the coagulation factors are going to be consumed in the clot. And then when we're testing for coagulation factors, we're not going to have anything, right? That's why we use plasma, okay? Plasma from sodium citrate, all right? So very good. Now, when we get a sample in the lab, what we do is we get it in the sodium citrate. One thing to keep in mind, and I think I'll talk about that in the next slide, is that it's going to have a 1:9 ratio. All right, so 1 part additive and then 9 parts blood. Okay, that is very important, and we have to always make sure that we fill the sodium citrate tube all the way to the little line that indicates that it's full, and that'll give us assurance that our tube has 1 part anticoagulant and 1 part blood. Okay, I mean, sorry, 9 parts blood. Okay, so when we get our specimen in Coag, we're going to centrifuge the specimen. Okay, and at the top you have the liquid part of the blood, and that is going to be called the plasma, and it has all of your coagulation factors. In the bottom, you'll have the red cells, okay? So here's a picture of the plasma versus serum, and I think this picture will help us understand. The plasma we collected in a sodium citrate tube. It's anticoagulated blood. When we centrifuge, the red cells go to the bottom, but then all of your Coag factors will go to the top of the tube. In a serum tube, you don't allow this. There's no anticoagulant, but when you spin it down, okay, some of your factors will get consumed at the bottom of the tube, okay. And when you allow it to clot, because the clot will form, okay. If there's no anticoagulant, right, the clot is going to form. And when this clot forms, it's going to bind things. It's going to bind red cells, you know, platelets, and then some of the coagulation factors. That's why they say that when you spin it down, the serum is actually going to be missing important coagulation factors because they're consumed in that clot, okay? So it's going to be fibrinogen, prothrombin, factor 5, factor 8, and factor 13 are going to be missing from that serum. So the testing that the tube that we use for Coag is the sodium citrate tube. Okay, so in Coag, we have names for the like grouping names for all of the factors that we talk about and that we test in Coag. Okay, and so these factors are grouped by their common properties. Okay, the fibrinogen group is fibrinogen, factor 5, factor 8, and factor 13. Okay, these all get consumed in the clot and they are acted upon by thrombin. Okay, the next group is the contact group. That is factor 12, factor 11, prekallikrein or PK, and high molecular weight kininogen. These are important factors for in vitro activation of the intrinsic pathway only. I know we haven't talked about the pathways. We will get into that in just a few minutes, okay? Activation of factor 12, prekallikrein, and high molecular weight kininogen is actually not important for the initiation of clotting in vivo, okay? So basically, in vivo, in vitro, right? We have two different things going on here. We have what actually is happening in the body as opposed to what is happening in a test tube, okay? For us to try to figure out deficiencies in factors, you have to kind of mimic what's happening in the body in a test tube, okay? That's how testing works. And so these factors, these contact group factors, factor 12, 11, pre-K, high molecular weight, they are not important for in vivo initiation of your coagulation cascade, but they are important for in vitro, okay? Which means that if a person has a deficiency in factor 12, PK, or high molecular weight, okay, this is not going to result in bleeding issues for the patient. It's just going to give me some prolonged test results, but it's not actually going to mean that the patient has a deficiency in bleeding. All right, is that clear for you guys? Do you guys understand that?
Yes.
Yeah.
Yes, perfect. Okay. All right, the other group is the prothrombin group. So that has prothrombin, factor 2, that's factor 7, factor 9, factor 10, protein C, protein S, and protein Z, okay? The important thing about this group, and you should put a big star next to this, is that they require vitamin K in order to be functional, okay? So I'm gonna explain this. So what happens is that during synthesis of these prothrombin group factors, which occurs in the liver, okay? This is the synthesis where it occurs. All factors are made in the liver. But for these in particular, we need vitamin K to be present, okay? Vitamin K catalyzes the carboxylation, all right, of the gamma carbon of glutamic acids in the GLA domain at the N terminus. I know it's a lot of these particular factors, okay? So gamma carboxylation of the glutamic acids results in the ability of the factor to be to bind to calcium. Okay, so you need that gamma carboxylation of the glutamic acids of these factors. Otherwise, they cannot bind to calcium. And again, what do we need calcium for?
Clotting.
We need for clotting, right? So we need the factors and we need calcium. And we need a phospholipid surface, okay, from the platelets, okay? So now what's happening is that vitamin K in the liver has to come in to do the gamma carboxylation of the glutamic portions, the glutamic acid portions of these factors, so that they have the ability to bind to calcium. Without this ability, they are not functional, okay? Which means that if there is no vitamin K, these factors cannot be functional. Now, do people sometimes have vitamin K deficiencies?
Yes.
Yes, absolutely. So how do you think that will affect these factors?
Won't be able to clot as well.
They won't be able to be functional, and they won't be able to participate in the coagulation cascade. So what do you think would be the ramifications of that?
Like vitamin K supplements?
Well, you can take vitamin K supplements, yeah, but what would be the issue if somebody is vitamin K deficient? Like, you know, before you find the cure, before you treat it, it would be that they have the inability to clot, right? So then there's some there's the possibility of bleeding. Okay, so this is what we have to keep in mind. So it's very tricky, right? We have to remember a lot of little nuances here. So these particular factors, okay, which I really want you to highlight with your highlighter, or I want you to make a big star next to this, are prothrombin, factor 7, factor 9, factor 10, okay? You know how I always remember these? Vitamin K dependent. I do 2 plus 7 equals 9. And then I remember that for some reason, I don't know why. And then plus 1 is 10, okay? So factor 2, factor 7, factor 9, and 10, plus protein C, S, and Z are all vitamin K dependent, okay? And so we need this gamma carboxylation that is happening through vitamin K in order to bind to calcium. Otherwise, they're non-functional. And calcium is necessary for the factors to form complexes on the phospholipid surfaces of tissue factor-bearing cells and activated platelets. It is important so it can participate in the generation of thrombin and the formation of the thrombin clot, which is the clot, okay? Another thing to mention is there are 2 sources of vitamin K, okay? Vitamin K is a fat-soluble vitamin, okay? And you can get that from your diet, all right? So you can get it from green leafy vegetables. You can get it from fish. You can get it from actually eating liver, right? And you can get it from the bacterial syntheses in the intestines, all right? So could it be possible that a patient can become vitamin K deficient? Absolutely, you can become vitamin K deficient. You can become, you know, depending on your diet, perhaps you don't take in too much vitamin K, right? Or also keep in mind, vitamin K comes from bacterial syntheses in the intestine, okay? So somebody can become vitamin K deficient if they are in a long course of antibiotics, right? Remember, sometimes when you take like some antibiotics for a long time, that could really mess up with the normal gut flora that we have in our stomach, in our intestines. And this bacteria that we have, that's normal, it's making vitamin K for us. So if we're in a long course of antibiotics, it could really mess that up, okay? So some people that are in these regimens of antibiotics could become vitamin K deficient. So it's something very important to remember, okay? That's we're talking about, you know, coagulation factors and vitamin K and all of that. Okay, let's see. Now, have you guys ever heard of warfarin? Maybe?
Yes.
Yeah, right? You've heard it. This is also called Coumadin. I should have said that. Coumadin, right? It's an oral anticoagulant, okay? And Coumadin or warfarin, we use it to treat thrombotic conditions, right? So these are conditions in which there is excess clotting, and we talked about these yesterday. You know, it's a condition such as a person going through or having a pulmonary embolism, right? Or a deep vein thrombosis in their leg. Okay, so the conditions like this, for conditions like this, a patient could be given an oral anticoagulant such as warfarin or Coumadin. So the way that it works is that warfarin or Coumadin is a vitamin K antagonist. It's a vitamin K antagonist, right? So we already know what vitamin K does, right? It's promoting the gamma carboxylation of the glutamic acid of these factors so that they can bind to calcium, right? And now when Coumadin comes in or warfarin comes in, it's an antagonist, so it's not gonna do that anymore. It's gonna stop it from doing that. So when warfarin is given to a patient, vitamin K dependent factors, okay, that are synthesized in the liver, right? They become synthesized still, but without that gamma carboxylation of the glutamic acids in the GLA domains, okay? So it's just stopping that gamma carboxylation. So it makes sense, right? It makes sense because it's an anticoagulant. It's Coumadin, it's warfarin. It's an oral anticoagulant. And what we want to do is we want to stop clotting. So it really targets those vitamin K dependent factors, which are which factors? Do we remember? Does anybody remember which factors are vitamin K dependent?
2, 7,
9,
2, 7, 9,
10.
10,
Vitamin C. No,
C,
protein C,
protein C.
protein S, and protein Z. Yep, there you go. So 2, 7, 9, 10, C, Z, and S, okay? So warfarin and Coumadin are really just targeting these vitamin K dependent factors. But these are important factors. So without them being functional, there is no clotting, okay? So that's something that we have to keep in mind. So these factors are synthesized in the absence of vitamin K. And so sometimes we call them acarboxy, okay? Or sometimes people call them proteins induced by vitamin K absence or antagonist. So they're called PIVKA, okay? Proteins induced by vitamin K absence or antagonist. And obviously they're non-functional because they cannot bind to calcium and they cannot form complexes on the phospholipid surfaces of the platelets. All right, any questions about that? All right, so this is a picture. Oh, do you have a question?
No, I don't.
Okay. So this is a picture of, it's right from your book. It describes the vitamin K gamma carboxylation of the vitamin K dependent factors. Obviously, as I was saying, warfarin or Coumadin comes in and prevents that gamma carboxylation. No, I don't need you to memorize this because it's a little complicated. I mean, honestly, I myself get, somewhat confused by this. I just know that, vitamin K is promoting the gamma carboxylation, and without it, this is not happening, and it's in the glutamic acid ends of these factors. So go over this briefly, but I don't want you to memorize any of this stuff right here, okay? All right. Okay, so in coagulation, we also have these complexes, these complexes that we have names for, okay? The 10-ACE complex, okay? 10-ACE, right, is a complex that activates factor 10 to activated factor 10. The prothrombinase is a complex that activates prothrombin to thrombin, okay? And when you have, again, that little a, okay, a little a next to the Roman numeral, that indicates that the factor has been activated. And I want you to always keep in mind that this is like a complement cascade. So that means that one factor is going to activate another factor that's going to activate another one, and then another one, then another one, okay? Until it stops in the fibrin clump, okay? So our whole point is we want to aggregate all these factors and these coagulation proteins on top of these platelet surfaces, right? That have been already aggregating so that we can stabilize our fibrin clot. So this was happening in the body, guys. This is what's actually happening. This complement cascades are happening. Okay. All right. So I just want to say, before I move on, that we have two different models in coagulation. We have a plasma-based model, and we have a in vivo model that is actually mimicking what is happening in the body. This in vivo model is also called the cell-based model of coagulation. Cell-based, okay? And then we have the plasma-based model, which is actually what's happening in the test tube, okay? As opposed to what's happening in the real body. All right, let's move on. All right, so here's where we're gonna get into the whole pathway thing and how it works and all of that, okay? So there's two different pathways in the plasma-based model. We're talking about the plasma-based model right now. And this is a way that we're trying to mimic what's happening in the body, but it's actually happening in the test tube so that we can test for each and one of these factors in the lab, okay? All right, so look, I'm gonna see if I can use my, can I just experiment real quick? Do you guys mind? I wanna use my whiteboard here. All right, let's see if I can, I don't know. I'm not too like techie. Yay, do you guys see this? I just made a red circle. Okay, cool. So good. I'm so excited. I can use this. Okay. So just let's keep it right here in the extrinsic tenase. Sorry, let me go back. Okay. So we're going to talk about two different pathways. Okay. This is again the plasma-based model. So just keep that in mind. Plasma. I'm just going to put plasma so you guys remember. Oh my God, this is so nice. Okay. All right, so let's do the two different pathways that occur in the plasma-based model. We have the extrinsic pathway right over here, and then we have the intrinsic pathway. We're gonna take it to the extrinsic pathway first, okay? Do you guys remember yesterday we talked about tissue factor? And we also said tissue factor is one of the first factors that is needed to initiate coagulation, the coagulation cascade, okay? Does anybody remember where tissue factor comes from? So tissue factor can come from an a subendothelial cell, right? Or it can also come from extravascular cells as well. Okay, so remember we talked about, you know, primary hemostasis where there is a break in the vessel, right? There's exposure of the subendothelium, right? And then there is collagen that has to be exposed. So in that whole process, tissue factor always gets expressed as well, okay? Because the body knows, right, that it needs it to start the coagulation complex, the coagulation cascade. So tissue factor is super important, okay? So again, it's gonna be a tissue factor bearing cell. It can be a smooth muscle cell, or it can be an extravascular cell. These cells are going to automatically express tissue factor. So you see that right up here. So the role of tissue factor is to automatically go and activate factor 7, okay? So as you see it right over here, right? So it forms a complex with factor 7. So it activates factor 7, right? And this is why you see the little a next to factor 7. And it forms a complex, okay? Now, this complex right here that has been formed between tissue factor and activated factor 7 is called the extrinsic tenase complex, okay? The reason why it's called the extrinsic tenase, it's because this complex activates factor 9, right? And factor 10 to activated factor 10, okay? So right over here, you see it. It activates factor 9 and factor 10 right over here to activated factor 10. Does that make sense so far? Let me know if you want me to go back, or if you don't understand something, or if I'm making too many circles. Just let me know. Okay, so on the surface of a platelet phospholipid membrane, okay, activated, so remember, this is a tissue factor bearing cell right over here, right? And this is a platelet right over here. This is a platelet. activated platelet, okay? So on the surface of a platelet phospholipid membrane, activated factors, factor 8, okay, and factor 9, okay, right over here. Sorry, I should have said there's an activated platelet up here. Activated factor 9 and activated factor 8 come together, okay, and they also form a complex, okay, and this complex right over here is called the intrinsic tenase. Again, this is a tenase because this complex will come and activate this over here, okay? So 10-ACE, intrinsic tenase, okay? So what happens, this intrinsic tenase comes and activates factor, also comes and activates factor 10 to 10A, which you will see right over here, okay? Now, the prothrombinase complex, okay, which is this right here that you're seeing, this is the prothrombinase complex, is it activated factor 10, okay, and its cofactor is activated factor 5 that's bound on a platelet, on the surface of a platelet, okay. This is called prothrombinase. It's called prothrombinase complex because it cleaves prothrombin to the active enzyme thrombin. So this is prothrombin right over here, okay, prothrombin, and it cleaves it to an active enzyme called thrombin. That's thrombin right over here, okay? So right over here, thrombin will have many different functions, but the main function is to turn fibrinogen over here into the fibrin clot. So this is the whole point of coagulation, is we want to form all these complexes to eventually form thrombin, because thrombin is going to go and activate fibrinogen to fibrin, okay? So this whole process right over here has a little bit more steps, and I am going to cover that in a little bit, okay? So is that, I know I've made a lot of things going on here, but did it help a little bit try to understand, or do you want me to do this again? Anybody? Are you guys still there? You're good?
Yeah.
Okay. So let me move on to this. This is kind of like a more simplified diagram. That this is also from your book, and it just shows you a more simplified coagulation pathway. Now we have the remember, this is the extrinsic part of it, right? Extrinsic, and this is the intrinsic pathway. Okay, so the extrinsic is always going to start with tissue factor and activated factor 7, and it's going to form a complex. It's called the extrinsic tenase because it's going to activate factor 10, okay? Now, the extrinsic, okay, is a little bit, I'm sorry, the intrinsic is a little bit different, okay? So intrinsic will involve the factor 9 and factor 8, okay, extrinsic tenase, and eventually that's going to activate factor 10, okay, then that's going to get together with activated factor 5, and that's going to cleave prothrombin to thrombin, okay. Thrombin then is going to form fibrinogen to fibrin, which is the fibrin clot, okay. And now just want to go back to this side over here, the intrinsic pathway. I don't know if you guys remember, but I did mention before that factor 12, okay, prekallikrein, and high molecular weight kininogen, right, are factors that are needed for the intrinsic pathway right over here, okay? But if you're missing these factors, it doesn't necessarily mean the patient will suffer from bleeding, okay? But these are the first factors that are needed to initiate the intrinsic pathway. So factor 12, prekallikrein, and high molecular weight are going to activate factor 11 right over here, which will in turn activate factor 9, and that will come together with activated factor 8, which will then form the extrinsic tenase which will then activate factor 10. That will come together with activated factor 5, and that will activate factor prothrombin to thrombin, okay? Okay, so we have two different cascades here. We have the extrinsic and the intrinsic. Again, this is your plasma-based model of coagulation, okay? I'm gonna keep talking about it so that it'll like pretty much get engraved in your brain, okay? So, any questions so far?
Not
Yeah,
so much.
I have a question.
Yes.
So at the top one, there's no factor 11 in the I'm sorry, I mean the previous model, this one.
Which one? This one that I have on the screen?
Yeah.
Yeah, it's just a diagram. It's not. It's just skipping. It's skipping to the factor 9 and factor 8. It's skipping to the intrinsic tenase complex that also activates factor 10 on the surface of a platelet. This model right over here, that's actually simplifying a little bit more because it's not really telling us if it's on the cell or the platelet, but just so that we can remember the sequence, I should say. But this is actually happening in the intrinsic pathway. You do need these factors, but this is the model for plasma-based model that's actually happening in the test tube, okay? It's happening in the test tube. I should have said that before. So you need these factors, these contact factors right over here to activate factor 9, and then activated factor 9 comes together with activated factor 8, and this is actually the extrinsic tenase that will activate factor 10, and then that will get together with activated factor 5, and then that activates prothrombin to thrombin, and then fibrinogen of fibrin. So this is more of the in vitro. This is the in vitro initiation of the intrinsic pathway, okay? In vitro initiation of the intrinsic pathway. On the other hand, let me just go back over here, is a diagram that is explaining the different coagulation complexes that form on the actual cell, what's happening on the cell, and what's happening on the actual activated platelet, okay? One thing to keep in mind too, and thank you for making me go back to this, is that remember I said we need that phospholipid membrane of the activated platelet, okay? On top of that, because that's where all the complexes form, right? And on top of that, we need calcium, okay? So calcium is always needed to be bound to the cell membrane so that coagulation, so these factors can bind. Calcium has to always be present. Calcium has to be present here and also here, okay? In this side right over here. And without calcium, the binding cannot occur. But this is a simplified form of the extrinsic and intrinsic pathway that is actually happening in vitro, okay? So we want to always be able to see this pathway as well, because this is what's happening in testing, okay? So if one of these factors were deficient or missing, coagulation cannot happen, okay? But thank you. That was a good point that you brought that because what does it mean? Because one diagram shows me something and then the other diagram didn't show me, the other thing. Okay, is that clear now or no?
Yeah, thank you. I understand.
Okay, cool. So, all right. So I'm just gonna go back and reiterate everything that I just said, explaining it in these in those pictures. And so, so now we're gonna go over the plasma-based coagulation model in vitro, okay, in vitro. It's happening in the test tube. So the plasma-based model consists of the extrinsic, intrinsic, and the common pathway, okay? So this is one pathway that I didn't mention yet, but we're gonna talk about that. So the extrinsic, whatever you saw on the left side of your screen, right, involves factor 7, factor 10, factor 5, prothrombin, right, which is factor 2, and fibrinogen, right? So the lab tests that we use in Coag, they're used to detect abnormalities in one of these factors. So either factor 7, factor 10, factor 5, prothrombin, or fibrinogen. The lab tests that we're going to use to identify these factors are going to be, the test is called the activated partial prothrombin time. Prothromboblastine time. It's called the APTT or the PTT test, okay? The intrinsic pathway, which is the other pathway that you saw, involves factor 12, pre-K, high molecular weight, factor 11, factor 9, factor 8, factor 10, factor 5, prothrombin, and fibrinogen, okay? So the lab test in Coag that we're going to use to detect the abnormalities in these factors is going to be the prothrombin time, the PT, okay? So the common pathway is where the extrinsic and intrinsic pathways meet, okay? So what they have in common are factor 10, factor 5, prothrombin, and fibrinogen, okay? So these are the common pathways. Okay, so let's talk about the extrinsic pathway, right? So it begins, as I said to you, with injury to the blood vessel, which would lead to the exposure of tissue factor, right? It'll be exposed in any tissue factor bearing cell. So tissue factor is actually released by injured cells that are not normally exposed to flowing blood, okay? So not normally exposed outside of the circulation. It can occur when there's a rupture in a blood vessel by plaque, all right? Sometimes plaque will do this, okay? The coagulation factors that are important in the extrinsic pathway are prothrombin, factor 5, factor 7, and factor 10. And these are the vitamin K dependent, okay? Extrinsic pathway. Activation of factor 10 is almost instantaneous when compared to the seconds or minutes required for the intrinsic pathway. So extrinsic pathway, or the tissue factor activated factor 7, will activate factor 10 immediately, okay? And it's a lot faster than the way it's happening in the intrinsic pathway. The intrinsic pathway begins when there is contact made, okay, when circulating blood or collagen is exposed to the subendothelial layers of an injured blood vessel. All the necessary components are found within circulating blood. That means there's, you know, the contact factors for the intrinsic pathway are prekallikrein, which is pre-K, high molecular weight kininogen, right? You have then factor 12, factor 11, factor 9, and factor 8. These are the factors that are part of the intrinsic pathway, okay? And this picture shows you nicely when there's a damage. I mean, this you already saw in your other lecture, but when there's damage to the subendothelium, I mean, to the blood vessel exposing the subendothelium, okay? And how platelets come in to adhere to the site of injury. Okay, this right here shows us the all three pathways, okay, of the plasma-based model of coagulation. You have your extrinsic pathway, your intrinsic, and then you have your common pathway. All right, the red part of this diagram is the intrinsic. Okay, it has the contact factors, factor 12, pre-K, high molecular weight, which are activated and then proceed to activate factor 11, okay? So factor 11, and then factor 11 activates factor 9 right over here, which comes together with activated factor 8, okay? Now, this over here means that there is a phospholipid membrane, okay? Activated phospholipid membrane, okay? So it is needed for this complex right here to begin, for this complex to form, okay? This complex over here with activated factor 9 and activated factor A, in turn activates factor 10, which comes together with activated factor 5 on the surface of the platelets, of activated platelets, which will then hit the common pathway, which is thrombin, right? Thrombin comes in and cleaves fibrinogen to form a fibrin clot, okay? Now, the way the thrombin works right over here is that when thrombin is activated, it has different functions. Thrombin can directly activate factor 13, okay? Factor 13 in turn then comes in and cleaves a fibrin polymer, okay, to make a fibrin clot, or thrombin can directly come in and cleave fibrinogen to form a fibrin polymer and eventually a cross-linked fibrin, okay? So we'll talk about the functions of thrombin in just a little bit. So again, the red portion of this diagram is your intrinsic pathway in the plasma-based model, okay? You need these contact factors right over here to start the cascade, okay? So activated factor 12, pre-K high molecular weight, will activate factor 9. Factor 9 will then, activated factor 9 will then get together with activated factor 8 on the surfaces of the platelet. That complex is going to activate factor 10, which will come together with activated factor 5. Factor 5 is a cofactor for factor 10, okay? In this whole process, you need the platelet phospholipids and you need calcium as well, okay? This complex right over here will activate thrombin, which will then turn fibrinogen to fibrin polymer to cross-linked fibrin. This is the fibrin clot right over here, okay? Fibrin clot. Oops. Now, on the other hand, you have your extrinsic pathway, right? This is the green part of it. The extrinsic pathway, what do you need? The first thing that you need is you need exposed tissue factor on subendothelial cells, okay? This exposed tissue factor will come and activate factor 7, okay? It'll then form a complex, okay? And then this factor 7 will come, this complex directly activates factor 10, which then will come together with factor 5, which then will activate prothrombin to thrombin, all right? Thrombin will then cleave fibrinogen to fibrin and form the fibrin clot. Now, the third pathway is right over here, which is the common pathway. That's delineated in blue, okay? This common pathway it's basically where both intrinsic and extrinsic come together. That's the common pathway. And that involves the activation of factor 10, okay, factor 5, prothrombin, and fibrinogen. So all of these in blue are part of the common pathway. Any questions about this diagram so far?
I have a question.
Mm-hmm.
For the 12A, the 12 activated factor, 3K and H and 40K, are they, what is it happening on? Like, is it happening on a platelet?
No, the contact, this contact group, you mean 12 pre-K high molecular weight?
Yeah, that one.
This is happening in vitro. This is happening in vitro. The plasma-based model shows you what's happening mimicking what's happening in vitro. So it's not happening like in the actual body, like the way it actually happens in your body, in a test tube. So in a test tube, there's a way to activate these contact factors so that they can initiate this intrinsic cascade, okay? Now, in the extrinsic form of it, what's actually happening on the platelet is going to be the activation of factor 9, which then comes together with activated factor 8, okay? It's gonna form that extrinsic tenase complex, and it's gonna need some calcium, okay? That complex is then going to, in turn, activate factor 10, which gets together with activated factor 5, which is its cofactor. Again, that's also happening in the activated membrane phospholipids, plus in addition to calcium, okay? And that in turn then activates thrombin, prothrombin to thrombin, which then activates fibrinogen to fibrin. So not this whole thing right here, no, not on the platelet. But once it gets to factor 9, yes, you need this activated, this phospholipid membrane to be there. So this activated platelet membrane, right, to be there, so plus calcium, so that this can continue to occur. This cascade can continue to happen. Okay?
Yeah.
Okay. All right, so now, so we talked about plasma-based, right? But for many years, the plasma-based model was the accepted model as the process of blood coagulation in the body. And that's because we just didn't discover enough, okay? So the model of coagulation that occurs in a this plasma-based model is a model of coagulation that occurs in the plasma in the absence of cells, okay? It's just like an in vitro model, something that we needed to come up with so that we can understand testing and understand conditions of coagulation. But through the work of Dr. Dr. Hoffman, which he first published in 2003, the cell-based model for coagulation was created, was basically out there. And it is now accepted as the process that occurs in the body. So it's the process that occurs in vivo, okay? So the model for blood coagulation gets initiated on tissue factor-bearing cells, but then it is amplified on the surface of platelets, okay? Now, just because we're going to talk a little bit about the cell-based model of coagulation doesn't mean that the plasma-based model is not important. Actually, the plasma-based model of coagulation remains very important because this is what we use to understand and to be able to interpret coagulation tests that are commonly used in the clinical lab, okay? So anything that has to do with in vitro testing is mimicking the plasma-based model, is the plasma-based model of coagulation. So the tests, the routine tests are the tests that I talked about before, the PT or the prothrombin time and the activated partial prothromboblastine time or the APTT. These are the two main tests. There's another test called the thrombin time. That test is also very important for coagulation, for understanding coagulation. All right, so I want you to keep in mind that the coagulation testing that we do in a clinical lab is just an artificial system, okay? And that doesn't include the total hemostasis process that actually occurs in the body, all right? It actually, this more information about this cell-based model can be found in the Fritzma factor. It is in the Fritzma factor website that you guys are going to go through a little later after this lecture. The thing that I also want you to know is that it's a very difficult process. The cell-based coagulation is probably beyond the scope of what you need to learn as an entry-level practitioner, as a medical lab scientist. So we're actually just gonna talk about it a little bit. I'm not gonna really go into it. This is more like a graduate level type of information, which you on your own time, if you definitely wanna look into this, if you wanna learn more about it, you should do it. You should really do it. But for us, in terms of what I'm gonna give you in a test, I'm not gonna go deep into this, okay? Because it's very complicated. Okay, but the plasma base, yes, we're gonna learn about the plasma base, okay? Wait, hold on, I'm sorry. Did I miss anything? No, I didn't. Okay. All right, so in this slide right over here, this is still from your book. I'm not gonna dive into this, but this is how the cell-based model of hemostasis actually works, okay? It is the in vivo physiologic coagulation process that is happening, all right? And that is always going to begin with tissue factor and activated factor 7. So factor 7 binds to tissue factor, and then it activates both factor 10 and factor 11, all right? So it's a very different process than what we're used to with the plasma base. So it comes in and it activates factor 10 and factor 11. So right over here and right over here, right? So you have your tissue factor varying cell, you have tissue factor being exposed, activated factor 7 coming in and getting together with tissue factor on the tissue factor varying cell. So these complexes are going to activate factor 10 and factor 11. Cell-bound activated factor 10 then combines with activated factor 5, okay? So cell-bound activated factor 10 gets together with activated factor 5, all right? And they generate small amounts of thrombin, okay? So here it is, thrombin. Remember I told you thrombin is very, very, very important? Okay, so thrombin has many different functions. Thrombin then comes in and actually activates platelets, okay? So right over here. It also activates factor 5, okay? It activates factor 8, and it activates factor 11, okay? And then that begins the formation or it begins fibrin formation, okay? Activated factor 9 is activated by both tissue factor and activated factor 7 complex, and also by activated factor 11. All of these combined with activated factor 8 on the platelet surfaces to activate factor 10, which then forms prothrombinase, right? The prothrombinase complex, and then produces a burst of thrombin right over here. So again, this is a lot more complicated, but this is what is said to be happening in the in vivo process. But again, I'm not going to be testing you on this, and I'm not going to be expecting you to learn this model, but know that the actual in vivo process takes into consideration the whole hemostasis process that's happening at the same time. It has different phases such as amplification, initiation, and propagation. Okay, it involves the promotion of thrombin, and it essentially both the cell-based model and the plasma-based model have one goal in place, which is to form a fibrin clot. Okay, so I do want you to keep that in mind. So just going back to thrombin, remember I told you thrombin is a very, very important protein, and I asked you to make a big circle around it. So thrombin is a key protease of coagulation, and because it has a lot of functions to it, okay? One function is it cleaves the fibrinopeptides A and B, okay, from the alpha and beta chains of fibrinogen molecule, which will trigger spontaneous fibrin polymerization. I'll show you a picture of that in a little bit. Thrombin amplifies the coagulation mechanism by activating cofactors 5 and 8, and also factor 11 by a positive feedback mechanism. Thrombin activates factor 13 to form covalent bonds between D domains of the fibrin polymer to cross-link fibrin and to stabilize the fibrin clot. Thrombin initiates aggregation of platelets, and bound to thrombomodulin, it activates protein C pathway, and that is used to suppress coagulation. And it also activates TAFI. TAFI stands for thrombin activatable fibrinolysis inhibitor. Okay, let me repeat that. TAFI stands for thrombin activatable fibrinolysis inhibitor. And that's used to suppress fibrinolysis. Okay, all right, so moving on. Let's start talking about the fibrin clot, fibrinogen. What is the structure of fibrinogen? And how does fibrin form, okay? And what is the function of factor 13, all right? All right, so let's talk about fibrinogen. Fibrinogen, as you can remember, as I said to you before, is synthesized in the liver, right? It has a very high plasma concentration, probably the highest of all coagulation proteins. I also mentioned that it is an acute phase reactant, right? So the levels of fibrinogen increase in inflammation, in cases of infection, and also other stress condition. We also said it increases in pregnancy, or when people take oral contraceptives, right? So any kind of stress condition the patient may be going through will have an increase in these acute phase reactant proteins, such as fibrinogen, okay? Now, the actual structure of fibrin, it consists of 2 parts, and it has 3 polypeptide chains, okay? It has the alpha, beta, and gamma chains, okay? The molecule forms a central E domain, and it has 2 D domains at the very end. So let me just show you. Right over here is the actual structure of fibrinogen. So fibrinogen has 2 parts to it, right? It has 2 parts of 3. So these are the 2 different parts, but it has 3 different chains. It has the alpha chain, the beta chain, and then the gamma chain right over here. All right. Excuse me. Fibrinogen is a molecule that forms a central E domain. So right over here in blue is the E domain. And then you have 2 D domains at each end. So this is the D domain, and this is the other D domain. All right. All right. So remember I said over here, going back, thrombin, right? One of its main functions is to come in, right? And right over here, it cleaves fibrinopeptides A and B from the alpha and beta chains of fibrinogen, okay? What this does is then it triggers spontaneous fibrin polymerization. So let me go back to that slide, all right? So thrombin comes in, okay? And it cleaves fibrinopeptides A and B, okay? Fibrinopeptides A and B right over here. So this is the function of thrombin on fibrinogen. It comes in and it cleaves right over here, okay? So it's only going to cleave the fibrinopeptides A and B of the alpha and beta chains, all right? So that's one of the main functions of thrombin, okay? Once it does that, it starts forming, like spontaneously, it forms a fibrin polymer, all right? Okay. So let's keep that in mind. Thrombin cleaves fibrinopeptides A and B from the alpha and beta chains in the E domain of the molecule. All right, so I said this before, right? Thrombin, which is activated factor 2A, binds to the E domain and cleaves 16 amino acids from the ends of the alpha chains and 14 amino acids from the ends of the beta chains, and it forms a fibrin polymer. The small cleavage products are called fibrinopeptide A and fibrinopeptide B. However, only the cleavage of fibrinopeptide A is required for fibrin formation. The fibrin monomer will then spontaneously assemble into a fibrin polymer. This is done via non-covalent bonding. The E domains spontaneously interact with the D domains, and then they form the fibrin monomers. The fibrin polymer, okay, which is also called the fibrin gel with non-covalent linkages, is the end point of clotting assays, okay? So let me just stop right over here to tell you what this means right over here, okay? The fibrin polymer is also called the fibrin gel, okay? the fibrin polymer will be formed from the fibrin monomers, okay? These little monomers spontaneously assemble to form this fibrin polymer, and this is all done by non-covalent bonding, okay? So they just spontaneously come in, interact, and then they form this polymer. Now, this polymer is the end point of clotting assay. So what this means right over here is that when we are detecting these clots in our coagulation systems, this is the last point that these clots can detect right over here. So this fibrin polymer, okay? It can't detect anything beyond that. So routine coagulation assays do not assess for factor 13, which I will get to in a little bit. All right, so after thrombin activates, thrombin, let me go back. So remember the functions of thrombin, right? So it cleaves fibrinopeptides A and B from the alpha and beta chains. It amplifies coagulation mechanisms by activating cofactors 5 and 8. It activates factor 13 to form covalent bonds between the D domains, okay? So now, thrombin, another special function right over here is that it's gonna come in and activate factor 13 to form covalent bonds between the D domains of the fibrin polymer to cross-link fibrin and to stabilize the clot. So that's another important function of thrombin, is that it's going to come in, okay, and it's going to activate factor 13 to 13A. Factor 13A, which is activated factor 13, will then form covalent cross linkages between the D domains of the adjacent monomers. And what this does is it strengthens the polymer and it renders it insoluble. So now it becomes a very strong stabilized fibrin clot. So this picture right here actually shows it nicely. And this is from your book. Again, you can always go back to your textbook. But this is the function of thrombin. So thrombin, which is activated factor 2, comes in and it binds to the E domains, right? It comes in, it binds to the E domains, and it cleaves 16 amino acids from the ends of the alpha chain, okay? And 14 amino acids from the ends of the beta chains to form a fibrin monomer. So right over here. So this is where thrombin is coming in, okay? Thrombin. So the small cleavage products are called fibrinopeptide A and fibrinopeptide B. So I want you to keep that in mind. Again, remember that we only need fibrinopeptide A for fibrin formation. The fibrin monomers then spontaneously assemble into a fibrin polymer. This is your fibrin polymer, okay? There's spontaneous polymerization. And that's done via non-covalent bonding, okay? The E domains spontaneously interact with the D domains of the fibrin monomers, okay? Keep in mind that the fibrin polymer is the fibrin gel, okay? And it is the endpoint of clotting assays. So routine coagulation assays do not assess beyond this point right here. This is the clot that the assays are actually detecting, okay? However, thrombin also functions to cleave or to activate factor 13, right? So after thrombin activates factor 13 to 13A, right, which is right here, factor 13A forms covalent cross linkages between the D domains. So you can see them right over here, right? Covalent cross linkages between the D domains of adjacent monomers. And what this does, it strengthens the polymer and it renders it insoluble. Okay, so it's now a stabilized fibrin clot. Stable fibrin, okay? So this is the actual clot, stabilized clot. This is the clot that is only seen by routine coagulation assays. All right, before I move on, you guys have any questions about this?
No.
No, we're good? Okay, perfect. Do you need me to take a break? Do you guys want to take a break? It's already 10:30. I know we're gonna like keep on going, but it's fine by me. I can keep on going unless you guys need a little break. You're good?
Yeah, we do.
Okay, perfect. All right, so moving on. Okay, so now that we learned about fibrinogen, we learned about the 3 pathways, we learned about the plasma-based model, and then we also learned about the cell-based model just a little bit. Now let's talk about some regulator proteins or regulatory mechanisms in coagulation. Okay, so just as much as we need these coagulation factors in our body to work, we also have to have mechanisms in place to regulate them, because we can't constantly have bleeding, clotting and clotting and clotting and clotting. We now also need systems in place to stop the clotting, all right? Okay, so antithrombin is a regulator, it's a serine protease inhibitor. It's a regulatory protein, okay? So it works in our body to stop coagulation in some sort of way, okay? So it's a serine protease inhibitor. That's also called a SERPIN, S-E-R-P-I-N, SERPIN, okay? It's produced by endothelial cells, but it's also produced in the liver. Antithrombin, when it's by itself, it neutralizes thrombin. It also helps in neutralizing activated factor 10, activated factor 9, activated factor 11, activated factor 12. Prekallikrein is also inactivated by antithrombin, but it's inactivated slowly, okay? So this is a protease inhibitor from the SERPIN family. Okay, so antithrombin is very, very important, and we're gonna always hear about this regulatory protein. Other inhibitors of the SERPIN family can be or are heparin cofactor 2, alpha-1 antitrypsin, alpha-2 microglobulin, alpha-2 antiplasmin, PAI1 protein Z dependent protease inhibitor, and protein C inhibitors. Okay, moving on. Antithrombin and heparin, in a heparin cofactor 2, both require heparin for effective anticoagulant activity, okay? When antithrombin is bound to heparin, it has a more rapid neutralization of thrombin. So it has a cofactor, so it needs this heparin cofactor 2 in order for it to work more effectively, okay? Heparin binds both antithrombin and thrombin, and it brings them together in close proximity, thus potentiates the neutralization action of antithrombin on thrombin. So basically, antithrombin, this regulatory mechanism, depends on heparin in order for it to occur, okay? Here's a picture of, this is again from your book, of how antithrombin works. Antithrombin has a heparin binding site, so it's right over here, okay? When antithrombin binds to heparin, the antithrombin undergoes a shape change, okay? In this shape change exposes a serine protease binding site, okay? This serine protease binding site needs to be exposed, all right? So antithrombin has it, but it has to be bound to heparin in order for this binding site to be exposed. Now, thrombin has a binding site for heparin, okay? So right here, thrombin always has a binding site for heparin. So when thrombin binds to heparin or to unfractionated heparin, it results in a bridging effect, okay? And that allows for covalent bonds to form between the antithrombin and the thrombin. So right over here, okay? Binding inactivates thrombin, okay? Antithrombin. So the binding that occurs between antithrombin and heparin, it'll neutralize thrombin. The complex is then released, and the heparin, or the unfractionated heparin, gets recycled. All right. Now, unfractionated heparin potentiates this antithrombin-thrombin reaction. All right. So we just need to keep in mind that antithrombin is a regulatory protein, and we need that to neutralize thrombin, but it works more effectively when it's bound to heparin. Heparin is another anticoagulant, okay? Here are some other regulatory mechanisms to keep in mind that are going to be important in the inhibition of coagulation. Tissue factor pathway inhibitor. Tissue factor pathway inhibitor binds to activated factor 10 and to activated factor 7 to prevent further activation of factor 10 by the tissue factor activated factor 7 complex or pathway. Protein C regulatory system is the activated protein C, which inactivates activated factor 5 and activated factor 8. Protein S is a cofactor for this process that binds and stabilizes activated protein C to then inactivate activated factor 5 and activated factor 8. C-dependent protease inhibitor, CPI, is a cofactor protein Z, which inhibits activated factor A, and it is a member of the serpin family. So I have some pictures here that I can use to explain that, those other regulatory mechanisms. Here you have tissue factor pathway inhibitor. Tissue factor pathway inhibitor binds to activated factor 10, okay? Right over here, sorry, to activated factor 10 so that it can prevent further activation of factor 10 by the tissue factor activated factor 7 pathway, okay? So remember, tissue factor, right, and the activated factor 7 complex, remember this complex? This tends to immediately activate factor 10, right? But this cannot happen, right, when tissue factor pathway inhibitor is bound to factor 10. This is why this is called a regulatory protein, okay? So the other protein, the other regulatory protein is your tissue factor pathway inhibitor. So, I mean, not the other, the same one, but this is how it works. I'm sorry. I meant to say the same, another illustration, all right? So here you have tissue factor pathway inhibitor, which binds to your activated factor 10 and activated factor 7 complex to prevent further activation of factor 10 via the tissue factor activated factor 7 complex. All right, so again, tissue factor pathway inhibitor is a regulatory protein that wants to prevent further coagulation. All right, next is the protein C pathway activation. Okay, protein C pathway activation. So after, the thrombomodulin thrombin complex. So basically here, we need, for this protein C pathway to occur, okay, we need thrombomodulin, okay? Thrombomodulin, after thrombomodulin, after binding thrombomodulin, thrombin activates protein C, okay, so right over here. This protein C is bound by endothelial cell protein C receptor. So it's a receptor on the endothelial cells. In this particular pathway, you need free protein S, okay, free protein S, which is a cofactor, and it binds and stabilizes activated protein C, okay? So free protein S binds and stabilizes activated protein C. The activated protein C, protein X complex, right over here, right over here, digests and inactivates activated factor 5, right? And activated factor 8 so that it can prevent further coagulation, okay? So this is, what this means right here is that it is inactivated. Inactivated factor 5 and factor 8. Now, this is occurring, okay? This activated protein C situation is occurring so that it can prevent further coagulation. All right, so again, the protein C pathway and activated protein C is a regulatory mode, it's a regulatory protein to prevent further activation. But for this pathway, you need thrombomodulin, okay? And that's gonna be exposed in the endothelial cell membrane. And you need protein C, which is typically bound to the endothelial cell protein C receptor, which is right over here. All right. For this pathway, you also need free protein S, which is a cofactor. Okay, so this will bind to activated protein C, which will then in turn inactivate factor 5 and factor 8. And again, all this is doing is we're preventing further coagulation. Okay, that's the way that the protein C pathway works. And this picture right here shows you the coagulation pathway with the regulatory proteins and where they come in, right? So you have the tissue factor pathway inhibitor, right? You have your antithrombin and where that comes in. You have your activated protein C and where that comes in. And also tissue factor over here. And you have the protein C dependence protease inhibitor. So with that said, with your now knowing your regulatory mechanisms, right, we can move on to fibrinolysis, which means we're breaking down the clot, okay? This is a process that has to be in place to break down the clot because we can't keep those clots. We have to get rid of them, all right? So the fibrinolysis is degradation of the fibrin clot. The fibrinolytic system is a way to break down the clot. You want to lyse the fibrin clot after it is no longer needed, okay? Physiological fibrinolysis occurs in the actual clot. Pathological fibrinolysis occurs when free plasmin is formed in the circulation. And there are conditions in which free plasmin can be formed in the circulation. All right, so plasmin, what is plasmin? Okay, so we're gonna cover that right now. All right, so for fibrinolysis to occur, we need activators and we need inhibitors. Okay, so the activators are tissue plasminogen activator or TPA. Has anybody ever heard of TPA before?
Is that
TPA.
used like for stroke patients?
Yes, very good. Yes, it is. Tissue plasminogen activator is an agent to break down a clot, okay? And it is actually physiologically something that we have in our bodies, but it can be synthesized to give us medication as well, all right? So tissue plasminogen activator, TPA, is an activator that we have to activate fibrinolysis, so to break down the clot, okay? The other activator is urokinase type plasminogen activator, or UPA. Both of these are released in response to inflammation or coagulation. All right, so I want you to keep this in mind. This can be released in response to inflammation or coagulation. So that means when we coagulate, the body knows it needs to break down the clot, so it's going to release these activators. At the same time, we also have inhibitors of fibrinolysis, okay? So inhibitors of fibrinolysis or anti-fibrinolytic systems are plasminogen activator inhibitor 1, PAI1, which prevents activation of circulating TPA or UPA and UPA, and we have the thrombin activatable fibrinolysis inhibitor, or TAFI, which prevents binding of TPA and plasminogen to the fibrin clot. So these are your activators, TPA and UPA, right? And your inhibitors of fibrinolysis are PAI1 and TAFI. Okay, so these are inhibitors of fibrinolysis, and these are your activators. Okay, so TPA and UPA. And again, yes, TPA is used to break down a clot in stroke patients. And there's a window in which you can do that, right? So plasminogen that binds to fibrin in the clots gets activated to plasmin by TPA or UPA. Plasmin then converts fibrin to fibrin degradation products. Free plasmin will rapidly degrade many proteins in the plasma. So if free plasmin is present in the plasma, it is rapidly inactivated by alpha-2 antiplasmin and or by alpha-2 microglobulin, okay? So these may also be called inhibitors, alpha-2 antiplasmin and or alpha-2 microglobulin. Okay, let me keep on going with fibrinolysis. So TPA, it is used to digest fibrin clots in acute ischemic strokes and early myocardial infarction and pulmonary embolisms. Deficiencies in plasminogen or plasminogen activators result in increased risk for thrombosis, okay, because you cannot activate fibrinolysis. Excessive fibrinolysis, on the other hand, can cause bleeding issues due to premature clot lysis before the wound healing has been established, right? So does that make sense? So now, does it make sense, like how these, we have these activators and these inhibitors, but if there's no balance, right, we can suffer from too much clotting or bleeding because we are degrading these clots way too early, all right? So this can be issues that arise. Okay, so moving on, this is a diagram of the fibrinolysis pathway. And also of your inhibitors, right? So we have our activators, which are TPA, right? And we have your inhibitors, which come in right over here. All right, so plasminogen and tissue plasminogen activator, plasminogen and TPA are bound to fibrin during coagulation. Okay, so you have TPA and your you have TPA and plasminogen. They're bound to fibrin during coagulation. TPA then converts bound plasminogen to plasmin, okay, which slowly digests the fibrin to fibrin degradation products. Okay, these fibrin degradation products are the X, Y, D, and E products, and also D dimers. D dimers are D fragments covalently linked together by covalent bonds. D dimers are produced from cross-linked fibrin. Okay, so that's what I want you to keep in mind. D dimers right over here come from cross-linked fibrin. Free plasmin is neutralized by alpha-2 antiplasmin, so right over here, okay? So we cannot have free plasmin going around, right? It has to be neutralized by a regulatory protein. TPA, on the other hand, is neutralized by plasminogen activator inhibitor 1, so right over here, okay? This is the neutralizing substance or protein. Thrombin activatable fibrinolysis inhibitor, or TAFI, right over here, is an inhibitor of fibrinolysis and inhibits fibrinolysis by cleaving lysine residues on the fibrin, preventing the binding of plasminogen, plasmin, and TPA. All right. So again, we might have to go back to this to understand it a little bit better, but as much as we have activators of fibrinolysis, we also have regulators of fibrinolysis, right? So TAFI, PAI1, and alpha-2 antiplasmin, these are regulators of fibrinolysis. UPA and TPA are your activators, but those are going to activate plasminogen and plasmin that is bound to fibrin. Okay, so again, plasmin will degrade fibrin in a progressive process, and that will form fibrin degradation products, or FDPs. And excessive amounts of fibrin degradation products in the plasma can actually interfere with the formation of a fibrin gel and inhibit clot formation. All right? Again, D-dimers are important because D-dimers are something that we try to detect in routine testing. D-dimers are the breakdown products of fibrin clot that has been stabilized by factor 13. There are 2 D portions and 1 E portion. Plasmin comes in and breaks down the fibrin clot, but it cannot break down its leftover fragment, which is the D-dimer, okay? So D-dimers are actually very unique products that result from plasmin breakdown of cross-linked fibrin only, okay? So when you see D-dimers, when D-dimers are positive, that means that fibrin has broken down cross-linked fibrin only, okay? D-dimers consist of 2 D domains that are held together by a covalent bond, okay? This picture right here shows you the degradation of fibrinogen and fibrin by plasmin, okay? So plasmin right over here, right? The degradation of fibrin, fibrinogen and fibrin by plasmin. Plasmin comes in and systematically degrades fibrinogen and fibrin by digestion of small peptides and cleaving the D and E domains. From fibrinogen, you'll get fragment X, which consists of the central E domain with the 2 D domains right over here. Okay, and then further cleavage by plasmin produces fragment Y, which is made of the D and E domains right over here. This eventually gets degraded to D and E domains. D and E domains right over here. Okay, so plasmin has this action of degraded fibrinogen and fibrin. Okay. Now, from the cross-linked fibrin, okay, fibrin comes in and its digestion produces fragment complexes from 1 or more monomers, okay? And D-dimers, right over here, right, consist of 2 D domains from adjacent monomers that have been cross-linked by factor 13A or activated factor 13A in a process of fibrin formation or thrombosis. So when plasmin comes in and cleaves this fibrin clot, what you'll see is the remnants D-dimers right over here. Remember, D-dimers are D domains that have been linked by covalent bonding. But when you see D-dimers, then you know that it's plasmin action on cross-linked fibrin only, okay? Cross-linked fibrin, okay? So this is the function of plasmin, okay? It'll come in and it'll degrade these into pieces. All right, so degradation of fibrin will result in fibrin degradation products and also D-dimers, especially when fibrin comes in and degrades the cross-linked fibrin, okay? Okay, I know that this has been a lot to take in. I know. Let me stop sharing. Okay, you guys still there? Good. Okay, hold on.
Yep.
screen broadcasting. Okay, hold on. Let me see. Oh, let me stop recording too, because